DNA structure and function. DNA stands for deoxyribonucleic acid. So in here on the right side we can see our DNA is a polymer and a double helix, and you have seen this already in GCSE. The DNA codes for the sequence of amino acid, and we've done this before: primary structure, secondary, tertiary, quaternary. This is the levels of structure in your proteins. The primary structure is that sequence of amino acid. In which order do you place the amino acid? That order is determined by the DNA code. So this is really important, because if you change the DNA code, i.e. mutation, you will change the sequence of amino acid. And then we have seen before that if you change the sequence of amino acids, you will change the shape of the protein, because you affect the bonds. In here I have a picture. We can see here these are marshmallows and spaghetti making different structures. The example that I want to tell you is that these marshmallows assume they are your amino acids, and these spaghettis making those bonds or connections, these are the bond that you have between the amino acid. Looking over here, this is our tertiary structure of a protein, and these are our amino acid. One amino acid here, one amino acid here, one here, one here. So proteins are made out of amino acids. So we are thinking of protein as our marshmallows. But there is a bond between the proteins, and these are not physical bonds, so they are between the amino acids, such as ionic bond, disulfide bond, hydrogen bond, and these are the bonds that make that unique shape. So just like if we rearrange the spaghetti's arrangement, and if you rearrange the marshmallow's position, you make a different connection, and that will change the shape. In the same way, if you start to rearrange and change this order and the sequence of amino acid, you will disrupt the bond and make a different kind of bond, and that will change the structure. And we know for enzymes this is so important, in particular the active site. The active site has a unique specific shape which is complementary to the substrate. So the active site here, if this is changed, then you will not have a substrate which will complementary fit into the substrate, the active site forming the enzyme substrate complex. DNA. So DNA is a polymer, and we've covered this before in biological molecule, talking about protein, for example. Protein is a polymer. It's got lots of amino acids. So DNA is also a polymer. So just like protein is a polymer made out of amino acid are the monomers. The monomers for a DNA that makes the polymer is called a nucleotide. So nucleotide is a monomer. In here I'm showing you a nucleotide. Yes, this nucleotide does have three different things. All of them together makes one nucleotide. Lots of these nucleotide, lots of these monomers will join together to form a polymer, and our polymer is called our DNA. For AQA, you need to know these three features of a nucleotide. So we have a phosphate group here attached to our sugar. This sugar has 5 sides. It's called a pentose sugar. The specific name is deoxyribose sugar. And attached to that sugar here, we have a base, and we've covered this in GCSE. We have 4 bases: adenine, thymine, guanine, cytosine. This base is a nitrogenous base. As you can see, it's filled with nitrogen. So to make DNA, to make the bases, you require lots of nitrogen. So going back to our sugar here, you need to make sure that you know the specific name: deoxyribose sugar. Pentose sugar is a general name. It just tells you it's a 5-sided sugar. Deoxyribose is the specific name. Just quickly going back to our proteins, the primary structure. So we know that we have 20 different types of amino acid, and all of the amino acid, they're exactly same, but what makes them all unique, the 20 different types, is the R group. The side chain is always different. In the same way, we have 4 different types of nucleotides. All of the 4 types are same. They have phosphate, they have sugar. Only this nitrogenous base will be different. Some of them will be adenine, others thymine, and so on. Now that we have done the nucleotide, the monomer, let's see how they make the polymer, the DNA. Polynucleotide, lots of nucleotides attached together, is a polymer, and this polynucleotide is also called our DNA. Over here I'm showing you one monomer. This is a phosphate attached to a pentose sugar, deoxyribose, and we have base adenine. Let's say another monomer, another nucleotide. There's going to be a bond. They will join together. This is a condensation reaction, so you're forming a bond between the sugar and the phosphate. So as you can see here, this is our bond there between the sugar and the phosphate. This bond is a strong covalent bond, and this bond is created between them, sugar and the phosphate. And you have to know this name of this bond, and this bond is called phosphodiester bond. And this is a condensation reaction, so water is released out. I'm showing you only 2 monomers linked together, 2 nucleotides, but you have lots of them joining together like this. So I have lots of nucleotide joined together. If you were to look carefully, the back side of this strand of DNA, this is one strand of DNA, the back side here looks like a bit like our backbone, the human backbone. So this is called sugar phosphate backbone. I have a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on. This is a sugar phosphate backbone, and you have to know this name. This sugar phosphate backbone, just like our bone, is very, very strong. And when it comes to this, why is it so important that DNA has a strong sugar phosphate backbone? We all know that DNA is 2-stranded. I'm showing you only 1 strand for simplicity. Let's do 2 strands. On the right hand side, I have 2 strands here. This is 1 strand, and that's the other strand here. So this is a DNA, a polynucleotide. That these 2 strands, they are not physically bonded together, but they have a hydrogen bond between the bases. And you have covered this in GCSE. The adenine will bind to thymine, and cytosine will bind to guanine. This is complementary base pairing. Between these 2 bases, there is a weak hydrogen bond, and that you need to know, hydrogen bond. And as you can see here, there's 2 lines there and 3 lines there, 2 lines there and 3 lines here. These lines represent hydrogen bond. So between A and T, there's 2 hydrogen bond. Between C and G, there's 3 hydrogen bond. These are our sugar phosphate backbone, one side and the other side. So what is the importance? Why is it so important to have a sugar phosphate backbone? This is a very strong sugar phosphate backbone because of the strong phosphodiester bond. This is to protect the DNA bases. These DNA bases, they are prone to chemical reaction. So we don't want them to go through chemical reaction and to be interfered and destroyed. The DNA is transferred from one generation to the next generation. You don't want any damage. You don't want any mutation between whilst you are transferring, while cell is dividing. So these sugar phosphate backbone, they make the DNA stable and they protect the bases from undergoing chemical reaction. Our next slide will have these information for you to write down. The second thing is these hydrogen bond between the bases. Another exam question would be, why is it important that we have, as you can see here, what's the importance of hydrogen bond between the complementary strands? This is a question that I want you to think about. I'll give you the answer now, but in the next video on DNA replication, it'll make more clear sense. The reason, because when you want to go through DNA replication, you have to separate these 2 strands. If these bonds were very strong, it'll be very difficult to separate. So making them having weak hydrogen bond, it allows you to separate the two strands with more ease. This question here, we can answer it full here. How does the higher proportion of C and G base pairing makes the DNA more stable? In our DNA, we have more of these C and G complementary base pairing. We have less of A and T. Why is that important? If you look into here, C and G is more, it's got 3 hydrogen bond. A and T is less, it's got 2 hydrogen bond. So if we have more of them, that means it's going to make our DNA more stable. So once we have formed our double-stranded DNA, one strand here, joined together by a weak hydrogen bond, it forms a double helix. These are the 5 points. This is how the structure relates to its function. We've covered, let's summarize here, sugar-phosphate backbone. And we know this backbone a strong covalent bond. The specific name is phosphodiester bond. Why is that important? It makes the DNA double helix a stable structure. It protects the chemically reactive organic bases which is inside the double helix. Weak hydrogen bond between the complementary base pairing, 2 strands. Why is that important to have weak hydrogen bond? Allows easy unzipping. So during DNA replication, when the 2 strands unzip, it allows that to happen with ease. That will be covered in the next lesson on DNA replication. Complementary base pairing. This is another important. We see A and T and C and G. They are complementary base pairing. This is also will be covered in DNA replication, but it ensures that the new copies of the DNA that are made are identical, are genetically identical. DNA is a large molecule. That's really important because that allows, that ensures that we can store lots of information, and it's double-stranded to allow replication to take place. We can replicate DNA because it's double-stranded. These number 2, number 3, number 4 will be covered in DNA replication.